And today let's learn something about Chapter 8, Adult and Aging Brain. The previous chapter described how your brain changes as you grow, in over size, number of cells, myelination, and synapse formation, even continuing to develop well beyond your teenage years. In fact, recent research suggests that maturation is still occurring in the third decade of your life. So, when does a human brain finally reach maturity? What is the structure of a fully formed adult brain? And what can it do that a developing brain cannot? First, the adult brain. An adult brain differs from an adolescent brain in many ways. Between childhood and adulthood, a human brain loses gray matter as excess neurons and synapses are pruned away. Although the rate of loss slows down by a person's late 20s, at the same time, some brain regions strengthen their connections with each other, and the major nerves tracts become wrapped in isolating myelin, which increases the brain's white matter. Around age 40, the white matter in the human brain has reached its peak volume. Much of the added white matter represents increased connections between widely separated brain areas. During childhood and adolescence, most brain networks are locally organized, with areas near each other working together to accomplish a cognitive task. In adulthood, the brain's organization is more widely dis- distur- distributed, with distant areas connected and working together. The most important brain area to become fully wired up in adulthood is the prefrontal cortex, the front portion of the frontal lobe. This area handles many of our higher level cognitive abilities, such as planning, solving problems, and making decisions. It is also important for cognitive control, the ability to suppress impulses in favor of more appropriate responses. Adult brains are better wired up for cognitive control than are adolescent brains, in which decision-making is more highly influenced by emotions, rewards, and social influences. Intelligence also peaks during early to middle adulthood, roughly ages 25 to 60. However, different cognitive abilities have distinctive patterns of maturation. Fluid intelligence, which includes abilities like solving problems and identifying patterns, peaks around age 30. By contrast, crystallized intelligence, which deals with vocabulary and knowledge of facts, increases until about age 50. Some scientists speculate that there is no single age at which all of our cognitive functions are at their peak. And this picture shows the brain matures. A fatty substance called myelin wraps around axons to speed up electrical transmission. This image shows axons wrapped in myelin, with exposed area in the middle called nodes of Renoir. And here is the problems and what is aging. There are some there are normal and pathological aging. And aging is a dynamic, gradual process, where it can be characterized by resilience in both physical and neurological health. Too often, aging increases the risk of injury and disease. One such risk is Dementia, a decline in cognitive ability that interferes with a person's day-to-day functioning. While aging is inevitable, dementia and disability are not. In fact, neuroscientists believe our brains can remain relatively healthy as we age. Pronounced decline in memory and cognitive ability, once thought to be part of normal aging, are now recognized as separate disease processes in the aging brain. Although the brain loses some neurons as we age, a widespread and profound loss of neurons is not part of normal aging. Nonetheless, some mental decline is normal. The continuous process of aging involves subtle changes in brain structure, chemistry, and function that commonly begin in middle life. Some studies suggest that cognition starts declining as early as the 20s and 30s, while other studies indicate that cognition improves into the 50s or 60s before declining. A growing area of neuroscientific research focuses on understanding healthy aging, which includes lifestyle choices such as diet and exercise, which support cognitive health throughout life. And how the brain changes. And there are several aspects about how the brain changes. First, cognitive changes. Subtle changes in cognition are a normal part of the aging process, with memory decline being the most common. However, not all types of memory are affected. Declarative memory declines with age, but non-declarative memory remains largely intact. As you learned in Chapter 4, declarative memory includes autobiographical autobiographical memory of life events, called episodic memory, and memory of learned knowledge, or semantic memory. Non-declarative memory includes procedural memory like remembering how to ride a bike or tie a shoe. Working memory, the ability to hold a piece of information in mind and manipulate it, also declines with age. Some studies suggest that a slow decline starts as early as age 30. Working memory, an example of the fluid intelligence mentioned above, is a set of cognitive skills that depend on rapid processing of new information rather than on stored knowledge. Other aspects of fluid intelligence, such as processing speed and problem solving, also decrease with age. Certain aspects of attention can also become more difficult as our brains age. For example, it may be harder to focus on what friends are saying when we are in a noisy restaurant. The ability to focus on a particular stimulus and filter out distractions is called selective attention. Another type of attention-divided attention refers to the ability to focus on two tasks at the same time. Activities that require this type of split focus, such as holding a conversation while driving, also become more challenging with age. And the second, structural changes. All of these alterations in cognitive ability reflect changes in the brain structure and chemistry. As we enter middle life, our brains changes in subtle but measurable ways. Studies using brain imaging techniques have revealed that total brain volume begins decline when people are in their 30s or 40s and starts declining at a greater rate around age 60. However, studies of individual brain regions suggest that the volume loss is not uniform throughout the brain. Some areas appear to shrink more and faster, than other areas. The prefrontal cortex, cerebellum, and hippocampus show the biggest losses, which worsen in advanced age. Several changes at the level of individual neurons can also contribute to the decreased volume seen in aging brains. The changes in aging are due to shrinking neurons, retraction and decreased complexity of dendrites, and loss of myelin. In contrast, the volume loss in adolescence is primarily driven by synaptic pruning and the death of excess cells. Our cerebral cortex, the wrinkled outer layer of the brain containing neuron cell bodies, also thins as we age. Cortical thinning follows a pattern similar to volume loss, with some regions of the brain affected more than others. Thinning is especially pronounced in the frontal lobes and parts of the temporal lobes. The temporal and frontal lobes are among the areas that demonstrate the most pronounced declines in both volume and cortical thickness. These are the areas that took longest to reach maturity. This finding has led to a last-in, first-out theory of brain aging, which holds that the last parts of the brain to develop are the first to deteriorate. Interestingly, studies of age-related changes in white matter support this hypothesis. The first of the brain's long-distance fibers to develop are the projection fibers that connect the cortex to lower parts of the brain and spinal cord. Fibers connecting diffuse areas within a single hemisphere, association fibers, are the last to reach maturity and show the steepest functional declines with age. And the third is neuronal changes. The aging brain also undergoes numerous changes at synapses. Although the synaptic changes are selective and subtle, their effect on cognitive decline is believed to be greater than the effect of structural and chemical changes. In the prefrontal cortex and hippocampus, scientists have observed alterations in dendrites and the branching processes that receive signals from other neurons. With increasing age, the dendrites shrink, their branches become less complex, and they lose dendrite spines, the tiny pro protuberance that receive chemical signals. A study in rhesus monkeys observes that the aging process targets a certain class of spines called thin spines. These small, slender protuberances are also highly plastic structures, extending and retracting much more rapidly than the larger mushroom class of spines. This has led scientists to speculate that thin spines might be involved in working memory, which requires a high degree of synaptic plasticity. The loss of thin dendrite spines could impair neuronal communication and contribute to cognitive decline. So far, direct evidence of their role in cognitive decline is lacking, and more studies are needed. Finally, the formation of new neurons also declines with age. Although neurogenesis was once believed to halt after birth, we know now of two brain regions that continue to add new neurons throughout life: the olfactory bulbs and the denated glyas of the hippocampus. Studies suggest that the rate of neurogenesis plummets with age in mass, but recent human studies suggest a more modest decline. It is not yet clear whether neurogenesis appreciably affects cognition in the aging human brain, but mouse studies indicate that strategies that boost neurogenesis can enhance cognitive function. And this picture above shows the synapses begin to weaken as a person ages, which can contribute to normal cognitive decline. And the fourth is chemical changes. The amount of neurotransmitters and the number of their receptors may also decline with age. Several studies have reported that less dopamine is synthesized in the aged brain, and there favor receptors to bind the neurotransmitter. Less robust evidence indicates that the amount of serotonin may also decline with age. And there is question about why does the brain age? The first speculation is that oxidative stress and DNA damage. DNA damage that accumulates over a lifetime could contribute to aging process throughout the brain and body. The DNA damage due to oxidative stress and received a great deal of attention. Every cell in your body contains organelles called mitochondria, which function a bit like cellular power plants, carrying out all carrying out chemical reactions that provide energy for cell use. Some of these metabolic reactions produce harmful byproducts called free radicals, highly reactive molecules which, if left unchecked, can destroy fat and proteins vital to normal cell function and can damage DNA as well. Your body has natural defense mechanisms to neutralize free radicals. Unfortunately, these mechanisms decline with age, leaving aging tissues more vulnerable to oxidative damage by the free radicals. Studies of the brain cells have shown that damage to their mitochondrial DNA accumulates with age. In addition, the brain of people with mild cognitive impairment and Alzheimer's disease show more signs of oxidative damage than the brains of healthy people. Studies in rodents also link increased oxidative damage to memory impairment. Your brain is one of the most metabolically active organs, demanding around 20% of the body's fuel. Its numerous Its enormous energy requirements make the brain even more vulnerable than other tissues to the metabolic changes that occur in aging. While the brain's energy demands remain high, its energy supply can no longer keep pace. The brain's ability to take up and use glucose diminishes and metabolism declines. And the second reason is immune dysfunction. Immune dysfunction often occurs in conjunction with the metabolic changes seen in aging. Microglia, the brain's resident immune cells, perform many important jobs, defending against pathogens, cleaning up cellular debris, and helping maintain and remodel synapses. These inflammatory responses are protective, but a prolonged inflammatory state is harmful to brain health. Microglia become more reactive with age, increasing the inflammatory response in the brain while also damping production of helpful anti-inflammatory molecules. Most studies suggest that excessive macroglial activity also contributes to cognitive impairment. The third reason is impaired protein recycling. We know that excessive buildup of abnormal proteins in the brain contributes to age-related neurodegenerative diseases like Alzheimer's and Parkinson's. Buildup of proteins and other cells components can also contribute to cellular degeneration in the healthy brain. Cells normally break down and recycle damaged proteins and molecules, using a process that is usually efficient but not perfect. Over time, damaged molecules can build up in cells and prevent them from functioning normally. Because neurons in the brain are not replaced as often as cells in other parts of the body, brain cells may be even more vulnerable to this buildup of damaged molecules. Also, the cellular machinery involved in breakdown and recycling process de- degrades with age, reducing the efficiency of the waste removal systems. Finally, remember that changes in the aging brain occur within the context of other changes throughout the body. Researchers speculate that worsened cardiovascular health, for example, would contribute to or even drive many changes seen in the aging brain. And that's all about the chapter 8.